Morning, dear students. Today we are discussing a very small topic, that is chemical digestion of food. Now, in the last class, some of you told me that it will be better if I can record some classes so that you can watch it later, or those who missed the class can also watch. So I thought I would record a few lectures. So along with the live interactive classes, some of the hours you can watch these recorded lectures. Okay. So the topic for the day is chemical digestion of food. So in the last class we discussed about the mechanical changes that the food undergoes as the food passes through the alimentary canal. And we know the major classes of nutrients which are present in our food: the carbohydrates, proteins, and the fats. Now, carbohydrates, they are basically complex polysaccharides which are present in our diet. Now, one example for this complex polysaccharide is the starch. Then cellulose is another example, but cellulose, we know it cannot be digested. It simply adds to the bulk of the food, and it is part of the fiber or the roughage. So it helps in the easy passage of food through the alimentary canal. The complex sugar that can be digested is starch. Then disaccharides like lactose, sucrose, maltose, etc. Now maltose, after digestion, it is converted to two molecules of glucose. Then lactose, the milk sugar, that is converted to glucose and galactose. Then sucrose by the enzyme, it is converted to glucose and fructose. So glucose, fructose, galactose, these are all simple sugars which can be easily absorbed into the blood. So what is the need for digestion? So that the food can be absorbed into the blood, and from the blood can be transported to various cells. So the complex polysaccharides, it cannot cross the epithelial barrier and it cannot reach the blood, whereas the simple sugars, they can cross the epithelial cell barrier and it can reach the blood. So that is the need for this digestion into simple molecules. So carbohydrates, they are complex forms. They are converted to simple sugar. The proteins are polypeptides. The polypeptides, they are converted first into short or smaller peptides like dipeptides and tripeptides, and then these dipeptides are converted to single amino acids. Then similarly, there are nucleotides which are acted upon by nucleotidases, converting it into nucleosides, which are further converted to sugars and their bases. Then fat, another main component of the food, fat. The fats are basically triglycerides. Now these triglycerides, they are converted by lipases into fatty acids and glycerol. So this is the changes. These are the chemical reactions that basically take place during the digestive process or during the chemical processes. These are the reactions that take place. So this is the digestive system of our body. So this is a very learned thing. You already know the digestive system. We have been learning this from very small classes. So the food first enters the buccal cavity. Inside the buccal cavity, in the last class when we discussed the Mechanical changes, we discussed about the chewing process, the mastication, and the swallowing, pushing the food into the stomach, etc. So here inside the buccal cavity, the food gets mixed with saliva. And the saliva contains, or the saliva is secreted by the salivary glands. The digestive system not only includes this duct, but it also includes the various glands which secrete the digestive juice, which helps in the chemical digestion of the food. So inside the buccal cavity, the main secretion is saliva. The saliva is secreted by the salivary glands. And there are three pairs of salivary glands: parotid, this is the one, then the submandibular and the sublingual. So these are the three pairs of salivary glands secrete the saliva. Then after the food gets mixed with saliva, it's after chewing, the food is converted to bolus. And then this bolus is then pushed into the pharynx. The pharynx, we know, it's a very short part of the alimentary canal, but it is a common part for the respiratory tract as well as the digestive tract. And pharynx is then followed by the esophagus. This is the esophagus, also known as the food pipe. And through the esophagus, the food is pushed down to the stomach. So here food rests for some time inside the mouth, gets mixed with saliva. Here some chemical changes take place. And then the food is pushed into the stomach. And inside the stomach, the food rests for a very long time. And a large part of the digestion takes place inside the stomach. Then from the stomach, the food is pushed into the small intestine. The small intestine has got a U-shaped portion known as duodenum. And into that duodenum, the leaf-shaped pancreatic juice is secreted from the pancreas. So the pancreatic juice also contains a lot of enzymes, and some digestion takes place here. And the remaining part of the digestion takes place inside this highly coiled small intestine, the ileum. And then here the digestion is almost completed and the absorption begins, or absorption takes place inside the small intestine. Then the remaining food is pushed into the large intestine. And inside the large intestine, water and some vitamins, they are all absorbed from the small intestine. And from the large intestine. The large intestine has got an ascending colon, then this is the transverse colon, and this is the descending colon. And this is followed by the rectum, where the fecal matter is stored, and this is the anus through which it is defecated. So this is the digestive system. Just to remind you, so this is the summary of the various chemical changes that occur during the process of digestion. So I told you we are discussing about the carbohydrate digestion, protein digestion, and the fat digestion. So here in the diagram, nucleic acid digestion is also shown. So the first, carbohydrate digestion. Now the carbohydrates are basically, chemically we discussed, they are complex polysaccharides. Now these polysaccharides, they are resisted by the enzyme amylase. So amylase is the enzyme which digests carbohydrates. The amylase is secreted by the, it's present in the saliva. So the amylase that is present in the saliva, that is known as salivary amylase. Now the amylase is also present in the pancreatic juice. It is secreted by the pancreas. So that is known as pancreatic amylase. So the digestion of carbohydrates begins inside the buccal cavity, and inside the buccal cavity the enzyme salivary amylase converts the polysaccharides into saccharides, smaller saccharides. Here it is smaller polysaccharides, it's actually smaller saccharides or simple sugars. And the here just the digestion just begins because the food only remains for some time inside the mouth. So from there the food is pushed into the stomach, but in stomach the amylase enzyme is not there. So the remaining digestion of the carbohydrates take place inside the small intestine. So here the major enzyme involved is pancreatic amylase. The pancreatic amylase convert the polysaccharides, smaller polysaccharides into disaccharides. And then again, the remaining part of the small intestine, these disaccharides are converted to monosaccharides. So that is how the digestion of carbohydrates is. So the two enzymes are salivary amylase and the pancreatic amylase. Salivary amylase, it is present in the saliva and that acts inside the buccal cavity. And pancreatic amylase acts in the duodenum because the pancreas open into the duodenum. It's part of the small intestine. And finally, these disaccharides are converted to monosaccharides in the small intestine by the action of these disaccharadases present in the intestinal juice. Finally, simple sugars are formed. Now, the second major category of food, the proteins. The proteins are polypeptides. Now, the polypeptides, they are digested inside the stomach. So, nothing happens to proteins inside the buccal cavity. The digestion of proteins take place inside the stomach. Now, in the stomach, the digestive juice that is secreted inside the stomach is called the gastric juice. So, gastric juice is secreted by the gastric glands which are present in the wall of the stomach. Then, the main enzyme that is involved in the digestion of proteins is pepsin. So, the pepsin is the protein digesting enzyme that is present inside the gastric juice. Pepsin converts proteins into small peptides, say oligopeptides. Then pepsin acts inside the stomach, but pepsin is normally present in an inactive form known as pepsinogen. So it is the pepsinogen is converted to pepsin when the food reaches the stomach. In the presence of HCl, this pepsinogen is converted to pepsin. So pepsin does its role and then the food reaches the small intestine. Here also the process is continued. So here the enzymes which digest the proteins are trypsin and chymotrypsin. So these enzymes are secreted by the pancreas. So they are present in the pancreatic juice. And this trypsin and chymotrypsin, just like pepsin, they are also present in the inactive form known as trypsinogen and chymotrypsinogen. So here trypsinogen is converted into trypsin by another enzyme known as enteropeptidase. So here this trypsinogen is converted to trypsin by enteropeptidase. Similarly, chymotrypsin is secreted as chymotrypsinogen and in the presence of food it is converted to chymotrypsin. So that is what happens inside the small intestine. So after the action of the trypsin and chymotrypsin, smaller polypeptides or small peptides are formed. Then these small peptides have to be converted into single amino acids. So that conversion is done by dipeptidases, carboxypeptidases and aminopeptidases. So they are present in the intestinal juice. So there are intestinal glands which are present in the wall of the intestine. So those intestinal glands secrete intestinal juice. That intestinal juice contains dipeptides, peptidases, carboxylpeptidases and aminopeptidases. So when we discuss the peptidases that digest the proteins, they are broadly classified as exopeptidases and endopeptidases. So exopeptidases, these are the exopeptidases. Exopeptidases act at one end of the polypeptide chain. So when they act at one end, amino acids from a terminal end is broken down. But there are endopeptidases also. These endopeptidases, they act at the middle or somewhere in between the long polypeptide chain, breaking the long polypeptide chain into short oligopeptides. Oligopeptides, they are short polypeptides. So there are endopeptidases, so pepsin, trypsin, chymotrypsin, these are all endopeptidases. So they break down the polypeptides into short peptides. Whereas the peptides which act down, which are present in the intestinal juice, like dipeptidases, carboxypeptidases, aminopeptidases, these are endopeptidases. Sorry, exopeptidases. They act at one end of the the short oligopeptides, and amino acids are formed towards the end. Okay, so that is about the digestion of proteins. So the protein digestion takes place inside the stomach and small intestine. It begins in the stomach, and there are endopeptidases like pepsin and trypsin and chymotrypsin. Trypsin and chymotrypsin, they are pancreatic enzymes. Then there are exopeptidases in the intestinal juice like dipeptidases, carboxypeptidases and aminopeptidases. Now we will move on to the fat digestion. Just before that, we will just see in brief nucleic acid digestion. The DNA, RNA, they are nucleotides. They are acted upon by DNA and RNA. They are acted upon by nucleotidases, converting it into nucleosides, and nucleosides are converted by nucleosidases and phosphatases into nitrogenous bases, sugars and fats. So we are now moving on to the digestion of fat. The fats are triglycerides. Triglyceride means 3 fatty acids plus a glycerol molecule. That is fat. And this triglycerides, they are broken down by the enzyme lipase. Now the digestion of fats take place in the small intestine. So nothing happens to fat in the stomach also. In the stomach, nothing happens to the fat, but fat digestion takes place inside the small intestine. So in the duodenum, that process takes place. It is done by the enzyme lipase. So the lipase is the key enzyme which converts fat into glycerol, fatty acids and monoglycerides. But there is one more digestive juice which helps in the digestion of fat. So we will see the fat digestion in a bit detail. So you all know which is that digestive juice. Bile. Bile is the digestive juice. It is the secretion of liver. So the liver secretes bile, and bile plays a very important role in the digestion of fats. So we will just see what is the role played by bile. So when the food is agitated means the peristaltic movements, the pendular movements, they all uh, take place and the food is mixed with the digestive juice. So here inside the small intestine, the food is after agitation. The food is mixed with bile, and during that time, so into the duodenum, liver secretes bile also. There are ducts from the liver secreting the bile into the duodenum, and inside the duodenum the food is mixed with bile, and the fat that is present in the food is converted to emulsified fat. So what is emulsified fat? Emulsified fat means, I just show you a diagram. So then that time I will explain it. So emulsified, first the fat is converted to the emulsified fat by bile, and it is the emulsified fat that is acted upon by the lipase. So lipase cannot act on fat simply, can only act on this emulsified fat. How the fat was emulsified? It is by the action of the bile, and this emulsified fat is converted by lipase into fatty acids and glycerides. So what is emulsification? We know the fats, they are fat globules, they are aggregates basically because they are not soluble in water. So during this mechanical agitation of the food, the large fat globules, they are broken down into small globules, but they can even rejoin again to form large aggregates. And the lipase, they can act only on the surface. So when these fat globules, when they break down into small molecules, the surface area is increased. So the lipase can act on all these areas. And here is where the bile plays the role. And when these fat droplets, they break down into small molecules, these are the bile salts. These bile salts come and bind to the surface of the fat droplets, or we can say these small fat droplets are coated with the bile salts. Then what happens is, when they are coated with bile salts, they will not stick to each other because their uh, non-polar ends are placed outside. So these bile salts will not stick together. We say it as the non-polar ends, which cannot, they repel, so they cannot attract, and so they will not again aggregate. So they will remain as small globules, and the lipase can act on the surface. So that is known as emulsification by the bile salts. So this emulsification is very essential for the lipase action to take place. So bile is actually playing a very important role in the digestion of fats. Now bile also plays another role that is known as micelle formation. Now, so along with the emulsification, so during emulsification, the fat droplets get coated with the bile salts. During that process, here you can see the hydrophilic part. Hydrophilic part, and the green part is the hydrophobic part. The hydrophilic part is faced outside, and the hydrophobic part is placed inside. And this is the hydrophilic part. So, and this hydrophobic part, it actually forms a very small chemical bond with this fatty acid inside, and hydrophilic part is placed outside. So this micelle, it actually helps in dissolving the fat in the water to a small extent. So the lipase, the molecules which are digested by the lipase, they remain in solution for some time before they are absorbed. So this is micelle formation by the bile salts, and during micelle formation, the bile it, it forms chemical bonding with the fatty acids inside. Then the hydrophilic part is placed outside, and it helps in keeping the fat droplets in the solution so that lipase can act. So for the action of lipase, bile helps in two ways. One is the emulsification wherein the fat droplets get coated with bile salts, and second one is the micelle formation wherein the bile it forms a temporary bonding with the fatty acids, and it make it partially soluble in the. Okay, so that is all about the digestion of fats. So we will just see in brief what are the things that we discussed today. So the chemical digestion of food you can categorize into the carbohydrate digestion, protein digestion, and the fat digestion. Now carbohydrate digestion, the key enzyme is amylase. Amylase is present in the saliva. It is known as salivary amylase. Then amylase is also present in the pancreatic juice. It is known as pancreatic amylase. Then the carbohydrate digestion, we can see it starts from the buccal cavity. It proceeds in the small intestine, and it is finished in the small intestine. Protein digestion, it takes place inside the stomach, or it starts from the stomach, is continued in the small intestine and completed. So here there are some very key enzymes like pepsin inside the stomach. Then there is trypsin, chymotrypsin. Then there are some exopeptidases which act at one end like dipeptidases, carboxypeptidases, and aminopeptidases, and converting the short peptides into amino acids. Then fat digestion, it is assisted by the pancreatic lipase, converting the triglycerides into fatty acids and glycerol. And there is the role played by bile. Bile helps in the emulsification of fats and also in the micelle formation, which helps in the digestion of lipase by the bile salts. So this was just a very brief topic, the chemical digestion of food, a continuation of the previous class where we discussed the mechanical changes of food. So when the food is mixed with the digestive juice, along with that some chemical changes also occur. That's what we discussed today, the chemical changes that take place in the digestive. So you can watch the video once again, take down good notes, and you can complete your assignment. Thank you.